Great. So welcome everyone. Thanks for showing up in the middle of the week. My name's Ollie, and I'll be sort of leading the way with Aubrey through this orientation to SNAPS, the Student Network for Amphibian Pathogen Surveillance. We're going to be recording this, and we'll also be distributing these slides after the fact, so don't feel like you need to be rigorously and vigorously taking notes throughout. All this stuff is going to come out in a bunch of different formats to you later. So please just follow along. Go ahead and go to the next slide. So just a quick overview of the stuff we'll be covering today. Number one, we'd like an opportunity just to meet each other. Oftentimes we're sort of operating in this vacuum and don't really know who we're working with. So it's great to kind of put some faces to the names of the people that we've been talking to up to this point. We'll spend some time talking about SNAPS in general, what the sort of goals of the program are, where SNAPS fits into the sort of larger framework of this North American BSAL task force, and what our sort of goals are in relation to the larger goals of that group. And also some background on sort of what SNAPS is trying to accomplish in the short term and more so in the long term, and where you perhaps could be more involved in SNAPS than you already are in terms of contributing to the sort of steering committee and group that thinks about some of our more long-term goals. We'll spend a good amount of time talking about the nuts and bolts of how SNAPS works, which I think is why most folks are here. Just sort of, how do I get the stuff? How do I sample it? Where do I send it to? All those sorts of ins and outs, and where you can get access to all those resources to remind yourself how to do that stuff, and some lesson plans as well. And then also where you can go for questions that might come up later as you get closer to the time that you're sampling. So we'll do some introductions. Mostly we just like to get to know your name, your pronouns, your institution and location where you are, the class you might be integrating SNAPS with, and the time period of sampling. And initially I was thinking about doing this as a big group, but it seems like we got a lot of people and I don't want to spend like 30 minutes doing this. So Aubrey, if you can maybe break us out into 2 or 3 rooms, and we'll come back in, 5 minutes or so, just so we can get to know a sort of subset of each other.
Okay, I am opening the breakout rooms now. So let's spend about 5 minutes and then we'll reconvene.
And we were involved in setting up this program and continue to support this program, and interested in seeing it growing.
I'll go second, since I'm sort of the odd person out here as well, in terms of I don't have a class. So my name is Michael Yabsley. I'm at the University of Georgia at the Southeastern Cooperative Wildlife Disease Study. And I'm one of the diagnosticians and parasitologists here. And so depending on where you are, some of your swabs may be coming to us at our diagnostic lab for testing. So some swabs go to the National Wildlife Health Center and some will come to us.
I'll go next. I'm actually kind of an odd person out as well. I'm a graduate student at Eastern Washington University, and I am in Dr. Jennifer Walk's lab, and she is a part of the working group for BSAL task force, which actually helped, kind of get all of this going. And I'm actually currently working on analyzing a lot of the SNAPS data that came in from the last year's, work that was done. And I'm working on analyzing it and hopefully maybe getting some good results from what happened. And hopefully it's going to keep going and growing from everything. So I'm excited to be here and learn what everybody else is experiencing and seeing what's going on. So yeah.
I'll go ahead. I'm Allison Sacerdote-Vallette. I'm curator of herpetology at Chicago Academy of Sciences Peggy Nodebart Nature Museum, and two of my wonderful coworkers from our education department are here if they want to say hi. Dave Bild and Abby Romo. And we're also kind of a little bit of a departure from the traditional model because we are based in a museum. So we're not working with college students. So if Dave or Abby wants to talk more about our group of students.
Oh, okay. So we have a teen program here at the Nature Museum. And so we get about a group of 16 teenagers from different schools all over the city in Chicago. And so they work with us to try out different field techniques and different field strategies and how to analyze data and think about all that. And so we're hoping to implement this and incorporate it with the program and have it be one of the things or activities that we do with them so they can learn more about it, try it out, and we can learn about it as well.
I'll just say hi. My name is Dave, pronouns are he, him, and Abby pretty much explained what we'll be doing. Nice to meet everyone.
I can go next. I'm Joanna Larson. I'm a researcher and assistant curator at the Museum of Biodiversity at the University of Notre Dame. I teach herpetology and vertebrate biology and do SNAPS activities with both classes.
All right, I can jump in if that's okay. My name's Aubrey Hill. I teach introductory biology classes at Tennessee Tech. The salamander skin microbiome has been my research focus for a long time, and BSAL surveillance has been a big part of that work as well. I joined the surveillance and monitoring working group just a couple of years ago, and just last month became the co-chair of the working group. So I'm really happy to meet all of you and looking forward to working with you this sampling season. And I think, let's see, Lenny, are you there?
I am here. Can you hear me? I'm Lenny Shiroz. I'm a wildlife biologist with the Canadian Wildlife Health Cooperative. And I'm coordinating the Canadian version of SNAPS this year. So I'm not lucky enough to be getting my own boots muddy, but I'm helping other people get out there and do their sampling.
Great. Okay, so unless anybody in this breakout group has a question for me or Mike or Lenny, then I guess we can go ahead and leave and go back to the large group. Okay, let's do that.
Nice to meet everybody.
Yeah, nice to meet you all.
This meeting is being recorded.
All right, I just gave the other breakout rooms 1 minute to wrap things up, so we should be all back in the large group session here in just a moment. I'm glad that worked. I was not really ready for the breakouts, but it worked well for us, so yay.
Good work.
It worked.
You never know.
You think after what, 3 years of doing these virtual meetings, I'd have it figured out, but no.
They keep changing it. That's the problem. You come in on Monday morning, you got something you need to do, and they've changed everything.
The life we've chosen for ourselves, right? Okay, so I believe all of our groups are back. Let me see. Oh, here we go. We got everybody back now. I'm going to go ahead and share the slides again with you all. For Are you able to see the slides?
Yes, we can see them well. Thanks for those intros, everybody. It was great to get to know the smaller group that I was with. I definitely learned a lot of really interesting stuff that I did not know ahead of time. So it's great, once again, to just kind of see the faces and connect it to what's going on at your home institutions. I did not get a chance to introduce myself and my group, but for you all, I'm Ollie Hyman, he, him. I'm at James Madison University, and I do this course with my freshman intro bio course. And you can see my students there. We went out to the field this last spring, and I'll be taking them out again in the spring. And I'll turn it over to Aubrey now.
Hi, everyone. I am Aubrey from Tennessee Tech University. Like Ollie, I'm a co-chair of the Surveillance and Monitoring Working Group. And today I'm going to be discussing the dual missions of SNAPS, which are surveillance and learning. So I'll start with surveillance. SNAPS sustains ongoing monitoring efforts for the salamander-eating fungus BSAL by utilizing the assistance of undergraduate students at colleges and universities across the continent. We currently have around 29 partnering schools and counting. We know that continued efforts to survey salamander populations will be critical for the early detection of BSAL in North America and a rapid response. And this is a huge continent. We can't do this alone, right? Therefore, we are hoping to scale up our surveillance footprint, and leveraging the energy, excitement, and far-ranging geographic distribution of college students is really what drives our success. And as new SNAPS partners, you all are an integral part of this, so thank you for your help as we expand our efforts. Now I'll briefly jump into our second major objective, learning. I'm sure you all know the benefits of hands-on experiential learning, especially when students can apply their newfound knowledge and skills to real-world problems. SNAPS learning objectives include disease literacy, biosecurity and aseptic techniques, awareness of threats to biodiversity, conservation measures, and more. Achievement is assessed through our pre and post sampling surveys, which we will discuss in more detail later. The surveys also include questions related to students' attitudes toward these topics and toward themselves and their own self-efficacy as young scientists. And our assessment results indicate that students have not only achieved these learning objectives, they also have positive attitudes toward conservation, fieldwork, and not least of all toward themselves. SNAPS boosts self-confidence and feelings of fulfillment because students know they've contributed to a good cause. They get excited about this, and as an educator, it really is a rare and wonderful thing to see that spark ignite. And in at least one case, I'm sure there's more, the SNAPS experience has encouraged students to get involved with more research, to present their findings at conferences, and even to consider a career path in a STEM field. One of my students, Xavier, who's pictured here, presented a poster about his SNAPS experience at the Global Amphibian and Reptile Disease Conference last year. SNAPS was his first field experience ever. GARD was his first conference ever, and this was his first research presentation. So as you can imagine, when he won second place, he was just so proud of his hard work and became inspired to present again at a local herp conference. Now he's hooked on herpetology and looking forward to grad school, and it all is thanks to SNAPS. So I think it has a much bigger impact on certain students than we may realize, which is a great thing. So to sum this up, SNAPS is a win-win situation for both the students and the salamanders. The salamanders are benefited by the students actually conducting the surveillance work. And in turn, the students benefit from the salamanders by getting this opportunity to participate in an experiential learning network and to engage in this real research project. And with that, I will turn it over to Mike Adams, I believe. He'll give you a breakdown of the structure of the BSAL task force.
Yeah, thanks, Aubrey. I wanted to just give a little bit of, I guess, context and background for the existence of SNAPS. It started, I guess, with a meeting that we put on at the it's the Powell Center for Ecological Synthesis, a USGS facility in Fort Collins, in 2015. Brought people in from 3 countries and all kinds of institutions, and we talked about getting ready for BSAL. BSAL had just been discovered in Europe, and we wanted to understand what we might be able to do to prepare for this pathogen that we really expected that we would actually discover it pretty quickly, but if not, that it would arrive in North America. And we're still concerned about that. And so what arose, one of the things that came out of that meeting was the structure that we call the North American BSAL Task Force. And this has a number of different components, but is sort of driven by a technical advisory committee in the middle that meets monthly and tries to coordinate a bunch of different things that are going on with preparation for BSAL. The working groups are listed there underneath, and those are sort of the main areas of activity. One of them is the surveillance and monitoring working group, which is the group that I guess oversees or originated SNAPS. SNAPS resides, I guess, as a product of that working group. And SNAPS, I want to say, is the only continental scale organized surveillance effort for BSAL right now. And surveillance is a really important part of this. We feel like early detection at this point would be key for the arrival of BSAL. We're trying to contain it and manage that. So it's really important in that regard. And you can just read these other working groups, I guess, but I want to mention, and I think others will mention, there's opportunities for involvement in both the surveillance and monitoring working group as well as other working groups if you're interested in that. But mostly, I just want to say that, we hope that you are involved in SNAPS because you find it valuable for yourself and for your students, but also know that this broader group really appreciates the work that you're doing, and it's important to this North American preparedness. It's also just providing data that's available to anyone to use in terms of just observations about both BD. Right now it's just zeros for BSAL, and we hope it stays that way, but it's also providing lots of data on BD occurrence. Thank you.
Just to follow up with what Mike was saying, so SNAPS really exists within this larger framework, and we're sort of a subgroup within this surveillance and monitoring group that's part of the BSAL task force. And SNAPS has 3 sort of major goals to contribute to the goal of that larger group. One is just to establish this ongoing surveillance program for BSAL and something that can be sustained in the long term. And this model seems to be working pretty well so far. We're also trying to coordinate the dissemination of all these surveillance results, so have them all in sort of one centralized location rather than individual labs sort of siloing the data in individual places and not having it all put together into one sort of larger picture that managers can work with. And then lastly, we want to, of course, provide a diversity of students with this engaging course-based research experience. So these are the 3 sort of major goals. And so I just want to talk a little bit, if you want to go to the next slide, about the actual sort of governance of SNAPS itself and how it works. So we have this sort of 2-year rotating position of leads where I'm on my 2nd year and Aubrey is in her 1st year. And so we keep a kind of conveyor belt of 2 different leads so that one can train the other in terms of what the general sort of responsibilities are. And then we have a lot of other contributing members. And mostly what we do is meet once a month just to sort of touch base on what our major goals are, what we're trying to get accomplished in this particular year, what things we have upcoming in the short term and what larger goals we want to accomplish in the long term. And the reason I'm telling you about that structure is because we always want people to come in and join this sort of contributing member group and get fresh blood and fresh ideas into the group. So please be thinking about that as you're implementing and doing SNAPs. So go ahead and go to the next slide. In terms of where we are with building SNAPs, at this point, we have a website, we've got a logo, we've got some lesson plans. If you've not had a chance to visit the website, we'll certainly send that link around afterwards, but we're working on building that up more and building more lesson plans. And we're now in our second big round of recruitment. That map to the right, basically all those little orange dots represent partner institutions and represents basically one of you that's going out and sampling with some of their snappers. And so right now we have 29 institutions. That's 23 in the US, 5 in Canada, and 1 in Mexico. But this is the people who have sampled this last year. This upcoming year, I think this is going to balloon to a few more institutions, with some more coming in Canada, a couple more people sampling in Mexico with area, and a few more people coming in the US as well. So this is our second big round of recruitment. You can go on to the next slide. In terms of where we are with having a central location to share data, AmphibiaWeb has this amphibiandisease.org website and this amphibian disease portal. And then within that, we have this sub project that is the SNAPS project. And basically all of the data gets funneled into this centralized database, compiled in sort of live figures that tell you the number of individual organisms of each different species that have been sampled. And this is actually downloadable as well. So you can download this as a data file just to sort of analyze it on your own or with your classes. And of course, managers can use those data as well. So this is the centralized database where everything goes. You can go on to the next slide. What I'd really like to communicate to y'all is sort of where we need help. So within this sort of SNAPS group and this SNAPS steering committee, we have several more even subcommittees, if you want to call it that. And those subcommittees are working on a variety of different things, like developing teaching and learning initiatives. So this could be things like developing lesson plans that are relevant to your course, whether it's, an upper division course or a microbial ecology course or a conservation biology course or an intro level course. We're always interested in seeing some of your lesson plans and developing those so that they can be distributed to the larger group, either through the website or through meetings like these. And also in assessing student learning outcomes, which is something that we're continuing and continually working on. We also have people working on the website and sort of social media and outreach and marketing in general. And I have the website in red there simply because that's something that we really want to sort of overhaul at this point. I think in general, we're of the mindset that the website just needs a lot of improvements. And so if there's anyone out there that is savvy with these sorts of things or is keen on working on these sorts of things, we'd love to get some input from you to sort of develop the website more. And also leveraging social media to get the word out about SNAPs. Another group is data management and research, which is basically establishing data standards, making sure that all this stuff gets entered into the database, trying to figure out what to do with all those data in terms of maybe a negative results roundup, a publication of some sort, the rules for data sharing. And also another big sort of problem that we're trying to deal with is there a way that we can incorporate data that's already being generated by other labs that aren't necessarily following our protocols into the SNAPS network? There's recruitment, chaining, and networking. So things like this onboarding session that we always need help with improving. And then another really big thing is, of course, grants and funding. We're always looking for new sources of funding. So any ideas that you all might have about ways to keep this going, we're all ears. And then, of course, any other ideas you might have in terms of expanding this between classrooms through paired classroom activities or maybe an annual student conference, creating webinars, or expanding to the pet trade or other pathogens. So once again, we are all ears. And if you want to go on to the next slide, we'll send around this link after the fact, but basically all this is just a basic survey of who are you and are you interested in being involved in which of these things, if any, are you interested in being involved in? So we'll send this around after the fact, but hopefully we can get some folks to join us during our sort of monthly meetings to talk about the bigger ideas.
All right, thanks, Ollie. To give you an overview of SNAPS and how all this works, I am going to take the lazy way out and play this pre-recorded video that Sasha Greenspan, who's here today, she made for last year. She did a great job explaining everything, and all of the info is still accurate and up to date, except for one little tidbit. The target sampling size has changed, so I will brief you all on that after playing this video. And just so you know, the link to the video is in the slide deck in case you want to revisit it later, need a refresher on anything. You'll be able to visit these slides in our handbook. They are posted. So here we go.
Hi everyone, this is Sasha and I will be doing an overview of how SNAPS works. So one of your best resources is going to be the SNAPS handbook, which is a Google Drive folder that you should all have a link to, and then we'll be sure to send around the link again after the meeting, just in case. A good place to start before field sampling is the onboarding checklist. This will take you through all the permits that you will need for sampling, as well as the learning assessment, getting your sampling supplies shipped to you, and also point you to a bunch of handy documents that we will go over in more detail later. Also, before field sampling, be sure to check out the lesson plans and learning assessment folder. Most of our lesson plans are still in the works, but we have a few resources to get you started. And we also strongly encourage you guys to come up with your own lesson plans and share them with SNAPS. And we really would love your participation on the learning component of SNAPS. Another great resource for you before field sampling is going to be our sampling guidelines and sampling protocol. So definitely read these over carefully, but I'll just go over some of the main points. So in our sampling guidelines, it'll first take you through how to select your species for sampling. So we've included a table of species that have been tested in lab trials for BSAL, and the species are organized into 4 categories based on vulnerability to BSAL, so highly vulnerable, vulnerable, carrier, and resistant species. So for SNAPS, we are targeting the top 3 categories, so highly vulnerable, vulnerable, and carrier species. We're avoiding resistant species if possible. And if the top 3, if these species in the table aren't found in your area, then taxonomically similar species are okay to sample. We also included a smaller table of species that we think are the most appropriate for student sampling because they are typically found in high abundance and easy to find in regions throughout North America. So definitely check that out for some guidance. Also in the sampling guidelines, you'll find some tips on how to select your sampling sites. So for the purposes of SNAPS, we define site as an area with uniform habitat characteristics. So when you choose your sites, we are targeting areas with high numbers of animals in the highest BSAL vulnerability categories, and we're prioritizing areas with high levels of human activity over more remote sites. For your sample sizes, we currently have funding for each institution to sample 2 species. 20 individuals of each species for a total of 40 individuals, but smaller sample sizes are still valuable if you aren't able to find 20 individuals of one species. The field sampling kit that you will receive from us will include swabs, tubes for your swabs, labels for your tubes, return shipping information and label, as well as an insulated box and an ice pack for shipping your samples back to us. Just a quick note on labeling your tubes, because it can be a little bit tricky at first. So when you go out sampling, you're going to be collecting 2 swabs for each individual animal. So you'll find that on the labels sheet, the stickers that will go on your tubes, there are 2 sets of labels with identical numbers, one with a red marker line through it and one without. So you will use the small circular and small rectangular sticker for your first tube. You'll use the larger rectangular label for your data sheet, and then you'll use the second set of labels for your duplicate sample tube. So after you labeled all of your tubes for each individual animal, you'll find that you have some extra stickers on your label sheet, and those are fine to discard. And you also don't have to label your samples in consecutive number order. They can go in any order. We've also included several swabbing tutorials in the sampling guidelines and protocol that you can check out to go over our swabbing protocol. And then within the SNAPS sampling protocol document, this will give you a list of materials that you'll need to bring out with you to sample. These are common field materials that you're likely to have in your lab already. It'll also go over the swabbing protocol. It'll go over how to collect your field control swab, what to do if you see sick or dead amphibians out in the field. It'll have a hygiene protocol for decontaminating your field equipment, and also sample shipment information. So after you finish sampling, you want to make sure that you do your post-sampling learning assessment with your students. And then for mailing your samples back to us, definitely see the protocol document for all the specific information, but just to go over the main points. In most cases, you will send back both of your duplicate sets of swabs, and Dan will go over this in more detail later. We ask that you organize your samples by site in plastic bags or in sample boxes within plastic bags. We ask that you include copies of your data sheets in your shipment, notify the National Wildlife Health Center before shipment, and the contact information will be in the protocol. And then we ask you to ship Monday through Wednesday so that your samples will arrive before the weekend. You'll also need to transfer your data from your hard copy data sheets to a spreadsheet that you'll find in the data sheets folder. And you'll email that spreadsheet to us at the time of shipment. So one of the big questions is what will happen if a sample tests positive for BSAL as part of SNAPS? So in the rules and policies folder of the handbook, you'll find a document that goes over a plan for this scenario. So this plan was based on the BSAL strategic plan rapid response template. And it'll explain that in the case of a positive sample, a core response team will be assembled, and this will include the SNAPS steering committee, some members of the BSAL task force, and other people who are going to be most knowledgeable in this scenario. And they will come up with planned for confirming the detection. If the sample tested positive at your site, then we would ask you to help in confirming the detection. And Dan can go over this in a little bit more detail later. The core response team will also come up with a containment and response plan, as well as a data release plan. So if the positive sample was found at your institution, then we would invite you to lead a manuscript to publish that finding. We ask for a 2-month timeline on the manuscript. And we also ask you to let us know of other authors from your lab that would be on that manuscript. Like if you had a graduate student or something like that that helped you over multiple years with SNAPS. So just wanted to let you all know that we are all here to help answer any additional questions that you might have. We also have an FAQ section of the website that you can check out, and just let us know if you need any help. Thanks a lot and have fun.
All right. Thanks, everybody. So as I mentioned before, Sasha did a great job explaining everything and how to find what you need before your sampling trip and after. I just need to update you on one little tidbit of information. So forgive me while I advance through the slides that Sasha just showed, and I'll share my screen again. The sampling size target has changed. So for 2023, we're now targeting 20 samples per researcher per year. So that would be 20 individual animals, meaning 10 individuals from 2 different species ideally, rather than a total of 40 animals. And this will just allow a little bit of funding for another school to participate in SNAPS. So hopefully that makes sense. And what Sasha mentioned during the video is that you have to collect 2 swabs per animal. And this is critical for several reasons, but importantly to confirm if a positive detection is found, we need that duplicate swab. So if you are able to capture 20 animals and then swab them twice per animal, this would be a grand total of 40 swabs. So hopefully this all makes sense, and we'll support you before you begin your trip and remind you of all these things. I just wanted to make sure that was noted before I turn things over to Dan Greer, who will let you know how the samples are processed.
Hi, everybody. I'm Dan Greer. I'm coordinate the diagnostic side of things with the USGS National Wildlife Health Center. We're in Madison, Wisconsin. So most of you will both receive the supplies from me and also send the swabs after successfully catching all the critters back to me. And that's kind of where I take over. The process really depends on a few steps. It's that once you get the swabs collected from the field, it's key that you send both the swabs along with the data sheets or the spreadsheet to me, and that will facilitate getting all the information, all the lab results run, and the information back to you for hopefully for use in your class if you want as quickly as possible. I can go into any details about the diagnostic procedures later, but we'll produce results for both BSAL and BD detection for each swab. Our aim is going to be, once we receive the swabs, to get them run within 2 weeks and get all that data quality controlled, sent back to you, as well as uploaded to the amphibian disease web disease portal project that Ali showed before. We'll do our very best to get them back promptly. Can't make all the promises to get them under 2 weeks, but if you have any scheduling considerations, like you're out near the end of the semester, or you'd really like data back for use in class sooner, please let me know ahead of time, and we'll do our very, very best to accommodate that. Because I think, although I have not directly been involved with any courses, some of the feedback, some of the most exciting feedback I've gotten is when students have received the results before the end of the semester and actually seeing what happens with their effort to go out and catch and swab the critters. So next slide. So just to get to the results, the bottom line is, as long as you send in the data spreadsheet along with the swabs, you'll get that returned to you again, ideally within 2 weeks. That will have all the information that you and your classes will need, but that data, and we'll take care of all of the data management and upload of the results to the amphibiandiseases.org project where we house all of the SNAPS data. And I think that's it. Try the next slide, Aubrey, and we'll see. And some time for questions.
Yep, so thank you so much, Dan. At this point, I'm sure you all have some questions, and Ollie and I will be happy to answer. And we also have other steering committee members here who may be able to help us as well. So feel free to unmute and or share a question in the chat if you would like.
All right, well, clearly we're just crushing it because no one has any questions. So that's either a very, very good, oh, there is one. All right, go ahead, Andrew.
Yeah, hey there. As far as this 2 species requirement goes, if we're able to capture, say, 10 newts and get swabs from them, but only 5 spotted salamanders, for example, is that data still useful to you, or do you want us to capture at least 10 samples per species?
10 is the suggested number, but a smaller sample size is still very useful to us. Let's say you're only able to capture a couple of spotteds or a tree frog or some carrier species that you happen upon. That would be great to just go ahead and swab it, even though the sample size is low. It's a data point and it could be useful. So yeah, and if you have leftover swabs, like let's say you get the 10 newts, but only 5 of the second species, maybe use the extra swabs that you still have in your kit to take some more samples from additional newts. And maybe Dan or somebody else can weigh in on this. But yeah, I say go for it no matter the sample size. It's just a suggested number.
Okay, great. Thank you. That helps.
Yeah, and if I could just weigh in on this, I feel like 20 newts is better than 10 newts. So if you know you're only going to get 10 newts and spend an hour in the field looking for something that you're never going to find, just swab 20 newts. But please step on my feet if I'm wrong about that, Dan, or anyone else.
Nope, that's right. It's as important for the students to get their hands on things as is getting some ideal distribution of swabs per critter. And someone asked in the chat if sampling frogs are okay too, and yes, it is.
Oh yeah, and just to sort of piggyback off that, please don't forget to get a group photo for the website. These have been pretty handy just to share on the website and just show all the different students and places that we're going. So please try to get some good group shots to share.
There's one more question in the chat, and it says if it is okay to sample frogs.
Yeah. Frogs are okay. I mean, ideally, you're kind of looking on that list to see what are the species that are susceptible and sticking with those groups or those species. It's probably something that in the future, maybe we need to be updating just as more information comes in about who and who is not susceptible. But in general, yes, I think frogs are okay. Were there a few more slides in there, Aubrey, about the assessment stuff?
Yep, sorry about that.
There's also another question in the chat about multiple sites.
So should all of the samples be collected from one site, or could we collect from 2 different sites in the same town? I think that would be acceptable, especially if you know one target species, a vulnerable species, for example, or susceptible species is located at an additional site near you, then yeah, that would be ideal. Hopefully you can just go to one site and get samples from 2 different target species, but if that's not the case in your area, then whatever works for you works for us.
Cool. Do you mind just pulling up those slides on the assessment stuff, and then we can wrap? Awesome, thank you. So the last part that we wanted to touch on is just the assessing student learning. Basically, we're trying to look at 2 major things with our students. Number 1 is sort of content related, like what do they learn about BSAL and BD and how pathogen surveillance can aid amphibian conservation, sort of field techniques and things like that. And then the other part of it is sort of related to what's called student affect and sort of how they generally feel about science careers in general, how they feel about how they can contribute to conservation and sort of larger scale things related to their future careers, and thoughts about how they fit into the sort of picture of science. So we'll also be collecting a little bit of demographic data just to get an idea of who our students' audience is and how we can maybe broaden into to more students and more groups. All of these data are anonymous and de-identified, so you don't have to worry about those sorts of issues. To go ahead and go to the next slide. To just sort of walk through the general process for how we're assessing student learning. Basically, you all just need to confirm with us that you plan to assess your students. This is all on the SNAPS logistics sheets that's been distributed by Sasha a ways back, but we'll ask you all again. Then we add you and your school to an umbrella IRB that we have through James Madison University, just so everything's done sort of above board. And then you'll get the link to share the survey with your students. So basically there's a pre-survey to kind of see where they're starting at, that you should do as soon as possible before they sort of learn anything in general about chytrid fungi, if you can avoid it. And then you'll do any SNAPS related activities. So this is usually like going out into the field. We know sometimes folks will do a sort of in the classroom type activity where they might download a database and kind of look at what the swab data are telling them. But whatever that SNAPS activity is that y'all do. And then there's a post SNAPS survey, and each of these surveys should take about 10 minutes. So if you're interested in doing that, and we highly encourage you to participate so we can collect those sorts of data, please let us know. And then if you want to go to the last slide, just to remind you where you get access to those surveys. So in that SNAPS handbook that we've been talking about a bunch of times, it's that shared Google folder. You'll see a link to onboarding. And then there's this 2023 SNAPS onboarding checklist. And if you go into that checklist, you'll see stuff about the SNAPS logistics spreadsheet where you can fill in whether you want to participate or not. And then there's the SNAPS learning assessment and links to the pre-SNAPS assessment survey and the post-SNAPS assessment survey. And I'll try to send around some semi-annoying emails just to remind you to do that stuff. So please be on the lookout for those. In terms of just contact information, as questions arise, please feel free to reach out to myself or to Aubrey. If it's stuff related to the sort of nuts and bolts of shipping swabs and sending stuff back and things like that, you can certainly ask me or Aubrey, but Dan and Megan are probably the gurus on those things, so you may want to reach out to them. And Sasha is also extremely knowledgeable, extremely helpful, but also has an entirely unrelated full-time job. So probably reach out to us first before her, but she's certainly helpful. So yeah, so that's all we have for now. We want to leave a little bit of time at the end for any questions or ideas that you all have. Like I said, we'll distribute a bunch of these sort of links to these documents and surveys and things like that shortly afterwards. And of course, a recording of this slide deck as well. But we wanted to spend, just a little bit of time for folks to just, ask any questions that you might have.
And it looks like there was a comment about reminding students to enter their anonymous IDs in the learning assessment, exactly the same in their pre and post surveys. So I guess that has been a little bit of a problem in the past. So it's a good reminder.
Yeah, that's a good point. And we've changed around the format of that anonymous ID to make it way more amenable to it matching before and afterwards. Before it was way too complicated. So hopefully it's a little bit easier now and that'll be fixed. But yes, that's important.
And then when to print the shipping labels. Dan, do you want to jump in there?
Yeah, so when we send you supplies, there will be I think a link, a hyperlink to FedEx to be to print a prepaid overnight shipping label. So you just print it and send it. But be sure to print that within a couple days, ideally, of when you're actually going to ship it, because they expire after a week or so. So when you're ready to go, ready to ship it, do everything all as in close amount of time as you can.
Okay, any other questions?
Other than the ice pack that comes in the shipping pack to return it, are there any other storage requirements for the tubes?
If you're not going to ship them immediately, freeze them in a
like if we
oh, go ahead.
Oh, I was going to say we're going to do our sampling on a Saturday, so if I can't ship them until Monday, just put them in the freezer.
Yep.
Okay.
Yeah, and try to avoid shipping on a Friday. And while you're in the field, it's usually good practice to just bring a little cooler out there, just something, and at the very least, just like try to keep it out of the sun and in a cool spot where it's not going to get cooked. All right, well, it's 12:57, so I guess I'm going to give you all a half an hour of your lives back. You're welcome. And I just want to say again, thank you all so much for participating. You're doing a huge service to the salamanders and to your students and to us. And just good luck and happy swabbing.
Yeah, thanks everybody. It was so great to meet all of you.
Thanks all.
Thank you.
Thanks.
Bye-bye.